recording. All right. So right now we're in the process of kind of getting oriented to what animal behavior is as a discipline. And so we've been talking about Tinbergen's 4 questions. We kind of finished that up last time. Before we go on and move on from that, how are you guys feeling about Tinbergen's 4 questions? If I asked you for the 4 questions and asked you what they actually were about, would you guys be able to tell me that on an exam?
Yeah, I think so.
Okay. What about the rest of you?
Yeah.
Okay.
Yeah, I also think so.
Okay. Right. I'm having some difficulties with remembering how to screen share on Zoom. I didn't really teach on Zoom all that much last semester. So give me a sec to get my screens doing their thing here. Can you guys see my screen? It's basically got the PowerPoint on it.
Yes.
Okay. All right. So after your textbook talks about, oh, hold on. I have to go and change everything. So basically, after they finished in the textbook, after they finished kind of talking about Tinbergen's 4 questions, they go into a discussion about the theory of evolution via natural selection. And this should kind of be review for you guys. So let's just see what you guys remember. Back in evolution and ecology, we talked about this. If you had me for genetics, which I don't think any of you did, but you might have reviewed this in genetics. Do you guys remember what the 4 preconditions are for evolution via natural selection to occur? And don't flip forward through the slide because the answers are there.
Within a nutshell, I think so. I have my book right in front of me, so it's all right
Okay.
here. Yeah, I definitely remember it from E&E.
Okay, so what's kind of the first one? And I usually take these in the order in which Darwin kind of covered them in The Origin of Species. And for each of these things, he, for the most part, devoted a chapter to each of these as he went through that particular book. What's the first one?
Individual variation within a species.
That's not the one that I usually have, but that's the second one usually. So we'll just go with that. So this idea that individuals vary, you can look at the screen right now and you can see, the 4 of us, and we are all different. Aside from the fact that I'm old and you guys are young, we have different hair color, different eye color, different builds. Everything about us is different. That's because we all had different parents giving us different complements of genes that then played out over our development. We were exposed to different environments. And as a result, each individual is different. A number of years ago, we had a pair of identical twins, and it was back when we had enough students in evolution and ecology that we had 2 sections, and they ran back to back. And in one section was one student who was the identical twin of another student. But even though they were identical twins, something about the way they went through ontogeny, the way they developed, made it such that I saw one of them in the 9 o'clock class, and then I saw the other one in the 11:15 class. I'm like, there's a Smith in the other class. Are you guys related? And she's like, we're identical twins. I'm like, really? I'm like, you don't look identical. you look like you could be identical, but you also look like you've undergone some divergence over the course of ontogeny, of development. So that was interesting. So they were identical twins. It's the first pair of identical twins that I could always tell apart, regardless of what they were wearing or what context I saw them in. So I generally think of the first one as kind of setting up what Darwin referred to as the struggle for existence. If all organisms in a population have everything that they need to survive, to reproduce, to raise offspring successfully, et cetera, then everybody will produce the same number of offspring, and that'll be the maximum number of offspring that they're capable of producing. And so this idea of Malthus from 1799, I don't remember how much we went into Malthus, but Malthus was this philosopher, but he was also something of a mathematician. He calculated the growth rate of humans in Europe and found that populations of humans, at least, were growing exponentially. And his prediction was that eventually humans would run out of food. There would be competition for resources. There would be overcrowding that would lead to plagues, et cetera, which meant that not everybody who was born was ultimately going to survive to adulthood and reproduce. And so this limitation of environments to be able to hold individuals of a particular species, the carrying capacity of an environment, places limits on who can survive and reproduce. And ultimately, that's the stage kind of on which natural selection plays out. Now, on that stage where not everybody gets to be fully successful, you have individuals that vary. Then what else do you know about this variation?
It has to be heritable.
Okay. And this is the one area where Darwin really didn't have very good ideas, because he wasn't aware at the time that the Origin was written, wasn't aware of Mendel's work, because Mendel hadn't done his work yet. It wasn't published until 1866, and Darwin published his stuff in 1859. But he did know enough about farmers, and he had been doing a lot of breeding of birds and things like this. He did know that, pardon me, he did know that traits were passed down from one generation to the next, because he had done his own breeding experiments. He had talked to a lot of farmers and talked about how they breed things. So he knew it was heritable. He just didn't know what the mechanisms were. So we have more offspring are produced than can survive. Individuals vary in terms of their phenotype. That variation is heritable. What's the last precondition that kind of brings the whole thing home?
There has to be competition.
Okay, so, and it won't always be competition, it can be predation or anything else, but that variation that you see among individuals has to have some sort of fitness consequence. So it could be variation that alters how well some individuals survive or not, how well individuals compete for limited resources, how well individuals avoid predation, how well individuals attract a mate, et cetera, et cetera. So the last one of these basically picks up this idea of individual variation leads to differences among survivals in either their leads to differences among individuals in either their ability to survive or their ability to reproduce. And so if you have all four of these things happening, then you will have evolutionary change via natural selection. You could have a population wherein more offspring are produced than can survive. Individuals vary among individuals. That variation is inherited, but if that variation doesn't lead to differences among individuals in their ability to survive or reproduce, then you won't have natural selection. In the same way, you can have a lot of selection, but if it's acting on a trait for which there's no heritability, then once again, you won't have evolution via natural selection. So all four of these have to be in place in order for evolution via natural selection to occur. And after we finish kind of going through this kind of overview of animal behavior as a field, we'll talk about evolution via natural selection and other forces of evolutionary change in more detail. But this is the basics of it. And the reason your authors mentioned this in the first chapter is that this is one of the pretty serious underpinnings of animal behavior. There's kind of a tacit assumption that behaviors evolved in response to natural selection. And we'll talk about whether that's a wise set of kind of preconceived notions to have about behavior or not. But that's kind of the driving idea that we adopt when we start looking at behavior. Behavior, in theory, should be adaptive. Behavior should help individuals reproduce, find food, compete with other individuals, find mates, raise offspring, et cetera, et cetera. Questions about those preconditions? All right, so your book talks about some examples and then doesn't really show you much about those examples. So one of the things that your book talks about is the importance of genes in behavior. Historically, there has been a long-running debate that you may have heard about, this kind of long-running debate about nature versus nurture. One of the things about behavior is that it is prone to phenotypic plasticity. Do you guys know what phenotypic plasticity is? Do you know what I mean when I say that term? You might have encountered it in genetics. Okay, nobody? Okay, so phenotypic plasticity is just a situation in which the same genotype will be expressed differently in different environments. So phenotypic plasticity is, oftentimes we quantify it as a genotype by environment interaction if we're doing an analysis of traits, but it's basically, you have the same genotype in two different individuals, you put those individuals in two different environments, and as a result of being in that environment, the environment influences what the phenotype is going to be. And so, for example, if you put an individual in a resource-rich environment, they might have a gene expressing one type of phenotype, but if you put that same genotype in a research-poor environment, it may express a different phenotype because of the differences in the resources that are available. We find oftentimes that individuals who experience stress early in life oftentimes shockingly have longer lifespan than other individuals. We could look at this in worms, for example, and look at the effect of stress on something like lifespan, and we might find that worms grown in a resource-rich environment have a different lifespan than genetically identical individuals that were raised in a research-poor environment. So behavior has long been recognized as being a plastic set of traits. They have this flexibility to them. As a result of that, there was less attention early on in studies of behavioral ecology. There was less attention paid to the genetic basis of behavior. And one of the studies that's mentioned in your textbook is the study on variation in these three loci for flies. And all of these things affect behavior. So Rover is actually a gene that is well known for affecting the activity of fruit flies. Then we have these other genes. I think this is actually a gene that that is a regulator. It's a why am I losing the name of it? It's a transcription factor. That's what I'm trying to think of. And so what happens in these two situations is when you have variation for Rover, it affects foraging behavior. And this foraging behavior is actually correlated with the activity of different enzymes. And so just having a dominant allele for the Rover gene makes you more likely to behave differently because it just changes your activity level. But just having allelic variation, just having a different allele for a particular gene isn't the only thing that affects behavior. Another study that, that your book mentions is a set of studies that have looked at bee behavior. What happens with bee behavior is really pretty fascinating. Bees, because they all the workers come from unfertilized eggs, they're all clones of one another. And so you have really low diversity among all of the sisters in a bee colony. So in the colony, you have a bunch of bees that are all kind of share the same genetics. They all got it from a single parent. And so what you have is you have different colony types. And in these different colony types, you actually have not different genes necessarily, not different alleles, but you actually have different levels of expression. So another way in which you can have genetic control over behavior is not by changing the alleles that are present, but changing how much those alleles are expressed. And so do you guys remember what kind of studies we do when we look at gene expression in organisms? Once again, you should have been exposed to this in genetics. All right, I'll take that as a no. So have you seen figures that look like this before, produced in a figure in a textbook or anything? These things basically relate to upregulation and downregulation of genes. So for a set of genes, you have typical colonies versus single cohort colonies, and you have young nurses and old foragers. So as the bees get older, they change their behavior. Young bees take care of the eggs. They feed them, they care for them, they get rid of fungi and stuff like this. As those bees get older, they shift their behavior and they become primarily foragers. And what we see is that for one suite of genes, the young nurses have upregulation of a set of genes, and all those genes are downregulated when they are found in older bees that are behaving differently. And for this set of genes, it's just the opposite. This is a suite of genes that in young nurses is actually downregulated, and in old foragers, these genes are upregulated. And you could actually blow this up and look at those genes, find out what those genes do, and see if you can make some functional sense out of whether or not those genes, relate to the kinds of things that would be necessary for foragers versus necessary for nurses. And so one of the ways in which we see behavior varying is just simply gene expression changes. Gene expression can change among environments. Gene expression can change, in this case, over the ontogeny of an individual, going from a young adult to an older adult. And so I think geneticists really have embraced this, and I think more biologists as a whole have embraced this idea that it isn't just allelic variation. Do you have a dominant allele versus a recessive allele that can influence the phenotype of an individual? If that allele doesn't get expressed, then that doesn't do anything to your phenotype. And so gene expression, I think more and more people are looking at gene expression in the context of behavior than they did, even 10 years ago in reality. Questions about gene expression? The way you measure gene expression is you expose individuals to a particular environment, or in this case, you collect individuals at different life stages, and you look at their messenger RNA. And just to remind you how this works, once again, I hope you were exposed to this back in genetics, but if you weren't, it's actually pretty fascinating. What you do is you collect the messenger RNA. Can you see that? Okay. Can you see that well enough? Or does it need to be bigger?
If you go speaker view and make the screen bigger, then you can see it pretty well.
Oh, let me see. How do I go speaker view?
I meant like just for us three. The students. You're fine.
Oh, okay. All right. You're telling that's instructions to those guys. Okay. Right. So I could also write slightly bigger, I suspect. So what you do is you get the cells and you isolate messenger RNA. So you basically isolate all the nucleic acids and then you essentially destroy the DNA and quickly then take the RNA that is in the cells and you convert that using reverse transcriptase. You convert that to complementary DNA, which is the complement of the DNA, which is the complement of the RNA rather. And so if a cell is activating a particular gene a lot, there will be lots of copies of the messenger RNA. So there will be lots of copies of the cDNA. Messenger RNA is very unstable. Do you guys remember why messenger RNA would be unstable? There are good biological reasons why you don't want messenger RNA hanging around for long periods of time in the cell. What would that be? Mason, are you saying something? No. Imagine that you are being attacked by a lion. And so all of a sudden you need a shot of adrenaline. The adrenaline that you have is a hormone, is produced in the adrenal gland. It's a protein. It's basically, it basically gets dumped in your system, partly because it's stored in the adrenal gland and it gets mobilized into the bloodstream. But you can imagine that you might need more adrenaline than what is actually stored in your adrenal gland. And so as soon as you start dumping the adrenaline in your adrenal gland into your bloodstream, the cells in your adrenal gland are going to start producing more of that substance. One, because you might still need it, and two, you need to replace what you've now gotten out of the adrenal gland and put into the bloodstream. And so, and I'm using adrenaline in the most general sort of way. I forget what the actual hormonal components of all of that are. So gene expression ramps up because you need to replace the adrenaline that is being depleted. But do you want to just keep on producing adrenaline? Once you've turned that gene on, what eventually happens? That gene that produces more and more adrenaline. You've escaped the lion, you've climbed up your tree, and
And you stop producing adrenaline?
yeah, you downregulate that gene. That gene stops getting transcribed into messenger RNA. That messenger RNA stops being used to produce more protein. But after you turn off that gene, after you downregulate that gene, because you're now safely in the tree, the lion can't get you anymore. If you have a bunch of messenger RNA molecules hanging around in your cells, what does your adrenal gland continue to do?
Produce adrenaline.
That continues to do that. And so messenger RNA is a molecule that kind of by design is ephemeral, because when you need a protein produced, you need messenger RNA molecules that will then serve as the template for making proteins. But as soon as you have all of that protein that you need, you don't want those messenger RNA molecules hanging around for a long period of time, because then you're just going to keep producing that protein when you don't really need it anymore. And so this is kind of the nature of gene regulation. You can upregulate and downregulate genes, but that upregulation and downregulation only really works if that downregulation then turns off the production of whatever the gene product is. So messenger RNA is notoriously unstable. If you've ever tried to isolate messenger RNA in a lab, it's very finicky. You try and isolate it and try and isolate it, and you never find it because it degrades very quickly. You have to keep everything on ice. You have to keep everything really, really clean. And so you want to try and get this messenger RNA into complementary DNA as quickly as possible. And you do this with reverse transcriptase. Now it's stored in a stable form, And then the number of copies of the cDNA you have, that's a measure of the gene expression. And so when you look at these graphs like you see on your screen, those are basically heat maps that show whether or not there's a lot of complementary DNA in a cell or a little bit of complementary DNA in a cell. And so you essentially count how many copies of the complementary DNA there are, and then that tells you what the gene expression was at the moment that you killed and destroyed those cells and extracted that messenger RNA. And once again, it's just becoming more and more commonplace to do these gene expression studies. Questions about that? Okay. Your book, since it's talking about natural selection, so these examples of genes affecting behavior were all because one of the preconditions of the theory of evolution via natural selection is this idea that inter-individual variation is heritable. If that variation is behavioral variation, then it's heritable and it can be influenced by the genes that are present, but it can also be influenced by how those genes get expressed. And then the last of the preconditions was this idea of selection. And notice that in this treatment of selection here, it talks about variation among individuals. And the reason for this is we generally view selection as acting on individuals. Individuals either live or die. Individuals either reproduce or fail to reproduce, or some individuals reproduce more than other individuals do. So this gets at this idea about this distinction between group selection versus individual selection. Have you been exposed to the idea of group selection in the past? And if so, what do you remember about it? Okay. Maybe one of the reasons you haven't heard about group selection is it's not really an idea that ever kind of survived in evolutionary biology. It was proposed by a guy named Wynne Edwards back in the 1960s. This is a situation in which selection acts to preserve the group, whether the group is a population of individuals, whether it's an entire species. And it's based ultimately in competition theory, because oftentimes it isn't just individuals that compete for resources. Sometimes it actually is groups that compete for resources, for example. And the idea is that individuals will reduce reproduction to avoid overpopulation and subsequent resource depletion. And so the idea is that if you have a population that is getting too big in terms of population size, you can reduce competition among individuals such that each individual has more food, can then have more babies, et cetera, et cetera. So you might reduce reproduction to avoid overpopulation and the resource depletion that will occur from that. What's the problem with that idea from a natural selection standpoint? It's a problem that we'll talk about repeatedly in this class over time. We have 7 individuals in the population. These individuals live in a really small geographic space. So to add an 8th individual would begin to deplete resources. And now all of these individuals are adults and they want to go and have offspring. If they each have 2 offspring, now you have 2, 4, 6, 8, 10, 12, 14, 16. 16 plus the original 8, that's 24 individuals where there were 8 individuals. Now you really have resource depletion. So now let's say that everybody kind of agrees to only have one offspring. That keeps the number down to 8, which was total of 16 because you have 8 parents, 8 offspring, keeps population down. And as a result of that, you don't have as much competition among individuals for resources. What's gonna get in the way of something like this evolving? What's the measure of evolutionary success of an individual?
Being able to reproduce.
What's that?
Being able to reproduce.
Yeah, being able to reproduce. And who wins the evolutionary game?
One that reproduces the most.
The one that reproduces the most. And so you can think about, pardon me, got something stuck in my throat today. You can think about evolutionary success at the level of the individual, or you can think about it at the level of the gene. Either way it works, but an individual is evolutionarily successful if it produces more offspring than everyone else. A gene is more successful if it can control its gene transmitter, the organism that transmits that gene into the next generation. A gene becomes more successful if it gets more copies of itself into the next generation. How does it do that? Well, it does it by coding for traits that cause the individual carrying that gene to have more offspring. This is a set of ideas put forth by Richard Dawkins in a book called The Selfish Gene. He was encouraging people to think about evolution, not at the level of the individual, but at the level of the gene. And in this case, he viewed the genes as the real actors in evolution, and the organisms possessing those genes were just tools of the genes to get them from one generation into the next generation. It's a productive way of thinking about that if you're going to think about this, because if you're a gene and you need to get from one generation into the next, and your success is governed by how many copies of that gene get into the next generation, any gene that causes an individual to cheat, it's going to leave more copies of itself to the next generation. As a result, even if everybody mates randomly, there's one more representative of that gene in the next generation. And so that gene is going to be more represented. It's going to get more represented in the next generation as well, because these two individuals will be producing more copies of that gene than any of these single individuals will be themselves. And so what you will generally find is that this gene that promotes higher levels of reproduction will spread through the population, because what matters evolutionarily is how many copies of your genes make it into the next generation. So this is where group selection, for the most part, breaks down. Fitness is the number of offspring produced. I've used this in a number of classes, so you've probably heard this. Who's the most fit individuals in the United States? At the moment. They're so fit, they have a TV program, or they had a TV program. I think they don't have a TV program anymore because there have been some scandals.
19 kids and counting or something like that.
What's that?
The 19 kids and counting or something.
Yeah, what did you say, Elizabeth?
I said the Kardashians. There's just a ton of them.
No, not the Kardashians. It's a different group, not so upscale.
I can't remember. The Duggars, is that?
Yeah, the Duggars. So the
Their last name?
Duggars. Yeah. I believe it's Duggar. 19 and counting. You guys know how many offspring Dr. Reynolds has?
I believe she has 2.
You are correct. She does have two. Do you know how many offspring Dr. Ron Lee and Dr. She have?
I think also two.
Actually, they had twins, so they're up to three.
That's right. I should know that.
They had twins. They had a boy, and then the next bout was twins. Dr. Vora.
I have no idea.
I believe he has three, two boys and a girl. Dr. Judd.
None.
None. Yeah.
None. Dr. Klowinski.
None.
None. The Duggars have 19 and counting. Dr. Ronley and Dr. Vora are doing better than Dr. Reynolds. Dr. Reynolds is doing better than Dr. Judd and I in terms of our fitness, but all of us are getting our butts kicked by the Duggars. So you can think of fitness. Avery, you're laughing. What do you have to say?
I just think it's a funny comparison sometimes.
So fitness can be measured in absolute terms, which is what we just did on the board, but you can also measure fitness in relative terms. And what you do when you calculate relative fitness is you take everybody's fitness and you divide it by the most fit individual in the population, the individual producing the most offspring. So we would, if we wanted to calculate relative fitness, the Duggars would have a relative fitness of 1 because they are the most productive individuals in the population. Dr. Reynolds would have 2 divided by 19. Lila and Joe and Bhupinder would have 3 by 19, and Dr. Judd and I would be 0 divided by 19. We still have a relative fitness of 0, but these relative fitnesses are all pretty small relative to the fitness of the top individual. Now, we could go into all sorts of discussions about whether or not this is good for American culture, et cetera, et cetera. It seems to me like smart, sensible people should be producing more offspring to increase those genes in the population. But this is in a sociology class. But that's how fitness works. Fitness favors those individuals that produce the most offspring. That means that those people are putting more copies of their genes into the next generation. And of course, what do you already know about some of these individuals in that set of 19 offspring?
That they're terrible people.
Well, once again, this is not a sociology class. I'm thinking more from the standpoint of fitness.
They have kids of their own, don't they?
Yeah, they've already started producing kids of their own. So these 19 offspring are going to ripple through the generations, because those 19 kids, what if they follow in their parents' footsteps and each of them produces 19 kids?
There's no way.
That's whatever 19 times 19 is, which 20 times 20 is 400. And at that point, like, that's a significant number of copies of those genes floating around in the population.
I couldn't imagine a family reunion.
Oh my gosh. You guys go to family reunions anymore? Oh, you still have them? Where's your family from, Elizabeth?
We haven't had one since COVID, but my mom's the youngest of 7.
Oh,
so
okay.
we're kind of spread out. I have family in Missouri, Kansas, and Wyoming.
Okay. Yeah, we used to have family reunions when I was a kid, but I think over time, as the generations got bigger and further apart, they just kind of fell apart over time.
Yeah, we just haven't had any since COVID, but we used to do them like at least 3 times a year.
Yeah. So group selection falls apart because group selection is ultimately destroyed by individual fitness consequences. And so that's the main thing you need to remember from that. So if fitness is the thing that really matters in evolutionary theory, why isn't everybody just producing 19 kids? Why don't we have superorganisms? Why aren't all individuals of all species performing at their maximum? So 2 questions to ask. Well, one question to ask about this. What do I mean when I talk about a superorganism in this kind of evolutionary fitness sort of way? What are some characteristics that would be of one of these superorganisms that would be the most fit, species on the planet? Most evolutionarily successful species on the planet. Think about it for a minute. The easy one I just erased.
So like it has the most amount of offspring that it could have.
Okay, so one characteristic of this evolutionary superorganism would be it has an infinite number of offspring. What would maybe some of the characteristics be of this offspring? What makes a successful offspring in terms of it being able to survive to reproduction?
Can you ask that again?
What are some characteristics of offspring that might make them better at survival? So you can produce a lot of offspring, that will increase your fitness. That doesn't increase your fitness if they all die. So what are some characteristics that would increase offspring survival, characteristics of the offspring themselves?
And them being like self-dependent, meaning like, I don't know, like lion when they have cubs grow, then I can't talk. Them being able to take care of themselves, I guess, like hunting and finding shelter and that sort of thing.
So in behavior, this is known as precocial. No, sorry. Yeah, precocial. Precocial offspring are offspring like ducks, where they hatch out and they're ready to go right into the water and start swimming around. Also correlated with what Mason just said, these offspring might be large. From the standpoint of reproduction, at what developmental stage should this superorganism that is like the evolutionary pinnacle, when should they mature and start reproducing their own offspring?
As quick as possible.
As quick as possible. If possible, they should be born sexually mature, because then they can just get right at the business of producing more offspring. Any other characteristics of these evolutionary superorganisms? This has to do with lifespan.
They would have a really long lifespan.
If you had a species that laid or gave birth to an infinite number of offspring that were all really, really huge, that were precocial, able to take care of themselves immediately, were sexually mature as soon as they came out of the womb or hatched out of the egg, and they lived forever, this species would take over the world. Why do we not? Can you think of any species that satisfies this set of characteristics? Nobody? That's a ridiculous set of characteristics, isn't it? Why? In theory, evolution should be pushing organisms to be these things, because all of these things result in more offspring. So what is it that evolution rubs up against that keeps this from happening?
Well, eventually you would run out of resources. Like your group would get so big that you can't sustain yourself anymore because you're like overrun.
But you would just become better competitors and screw all those individuals in your population who couldn't compete. They would just die of starvation and you would be sitting on top of the world.
Wouldn't the superorganisms compete with themselves?
Sure, but once again, it's like, it doesn't matter to me if other members of my species are dying, as long as I've got enough.
Yeah.
It's a very Darwinian way of thinking about things. Social Darwinism is what it came to be known as. What happens when you have a lot of offspring? When you have a lot of offspring, what else up here changes?
Aren't they usually smaller if you have a lot of offspring?
Exactly. If you have a lot of offspring, the size of the offspring say big and small. If you have very few offspring, you can take all that energy that you have for producing offspring, you dump them into a single individual, that individual gets to be big. But you can think about the energy that you have for reproduction as kind of a finite box. And so you have a box of energy. If you don't divide that box up at all, then that one individual offspring gets all of the energy. So it's going to be a really big individual. Split that up between two individuals. Now those individuals have to be smaller because that energy that you have for reproduction is divided in half. Have four individuals, that energy that you have for reproduction is now divided among four individuals. Keep dividing, keep dividing, keep dividing, keep dividing. The more offspring you have, the smaller those offspring have to be, because you don't have an unlimited supply of energy to devote to reproduction. So we generally see what's known as the size number trade-off, where there's a negative correlation such that species that have a lot of offspring produce very small offspring. Individuals that produce large offspring necessarily have to produce fewer of them. Why do individuals not have an unlimited amount of resources to put into reproduction? This gets back to some of the things you've mentioned in the past.
Because they have to have energy and resources to put into other things, such as like taking care of themselves.
Okay, so I drew this box of energy up here. This was the reproduction box, but reproduction isn't the only thing that you're doing. You also have to have some energy for what's generally known as growth and maintenance. And this can include growth and development, things like this, but it can also include maintenance, like just keeping you alive from one time unit to the next time period. And that can be energy put into avoiding predation, et cetera, et cetera. You also have to put energy into reproduction that isn't actually being dumped into offspring, but may be put into reproduction in terms of parental care or advertising for mates or any of those precursors to the actual production of the offspring. So you have a certain amount of total energy. Whatever you put into growth and maintenance can't go into reproduction, and vice versa. You can't double dip. Energy that goes into reproduction only goes into reproduction. Energy that goes into growth and maintenance only goes into growth and maintenance. So this gets to the general idea of constraint. The number of offspring that you have then constrains the size that those offspring can be. So you can't produce a large number of offspring that are really, really big. There are some organisms that do produce a lot of offspring in one big reproductive bout. And because they're putting all of their energy into this one big reproductive bout, after which they die, these species are called simulparous species. These are species that breed once in their entire life. And so they spend all of their lives growing up and getting big. And then after they've reached, in many cases, this huge size, this huge body size, they dump all of their energy that they have into reproduction, and then they die. Which basically means that they're saving no energy for growth and maintenance, because they're taking all of that growth and maintenance, putting it into reproduction, which then allows them to have a lot more offspring. So there are different strategies for accomplishing getting offspring, but those strategies are always balanced by these constraints on how much you can actually reproduce and what those offspring actually look like. So the answer to why don't we have superorganisms is because there are constraints in the system. Organisms don't have an infinite amount of time, energy, etc., to devote to reproduction. And so you find lots of different ways of going about this, because lots of different species have found different ways of kind of balancing these constraints. In human beings, we have relatively few offspring. That takes a long time to gestate them, 9 months. After that, they're not particularly precocial. We have to spend a lot of time in parental care to get them to the point that they're kind of self-sufficient. And so we put a lot of investment, a lot of energy and time into producing offspring. And as a result, we don't have very many offspring. A praying mantis, you guys have seen praying mantids around here. They're pretty common here in Missouri. They lay a big egg sac. And when those babies come out, there are hundreds of them, 400, 500 of them in a single egg sac from a single female. But they're all really, really tiny. You don't notice them a lot of times when they're really, really tiny. You notice them when they're big and adult. But if you find an egg sac, you can bring it in. I find them often at kind of the end of the season. Oftentimes, if you get a live Christmas tree, you'll actually find praying mantis egg sacs, egg cases in the trees. You can cut those off, save them in the spring, put them in a moist container, and you can hatch out the babies. And what you'll find is they're really, really, really tiny. That individual has taken a bunch of energy, put it into a bunch of little tiny offspring. Those offspring are self-sufficient, even though they're very, very tiny. But what happens to most of them?
They die.
They die. And so a praying mantis is taking a different strategy towards reproduction than a human is. A human is putting all of this energy and time into making sure that that one offspring that they have doesn't die. A praying mantis is playing a different numbers game. It's putting its energy into hundreds of little tiny babies that are self-sufficient, but have a very low chance of survival, in the hopes that one or two of them actually make it to adulthood. I mean, if a praying mantis could hope, which we don't know that praying mantids do hope. So different strategies, different approaches to get to the same outcome. A praying mantid may produce only one or two offspring in the same way that a human may only produce one or two offspring, but they get to that one or two offspring through totally different paths. This illustrates that when we look at the clutch size of individuals. This is the distribution of clutch sizes. I think these are for great tits in England. Paris major is the species. You see that there's a range of clutch sizes in terms of numbers of offspring, 5 offspring all the way up to 12 offspring. But then when you look at the number of clutches that they can have, the individuals who have relatively small clutches have quite a few clutches. And if you start increasing the size of the clutch, you start to bite into how many clutches you can produce over the course of a year. Once again, this idea of constraint. This is the size number trade-off. Once again, this is for those same birds. These large clutches they produce a lot of offspring in them, but these offspring are all relatively small, and the smaller clutches have considerably larger offspring. Once again, because these birds have only so much energy to put into reproduction. You can divide that up into a large number of small offspring or a small number of large offspring. So this illustrates this idea of constraint. Clearly, evolution has crafted some optimal clutch size that allows you to get a good number of offspring per clutch, and also allows you to have the number of clutches, a large number of clutches over your lifespan. If you deviate one direction or another direction from this, your fitness ultimately goes down because you're behaving in suboptimal ways. And the idea, once again, in animal behavior is that evolution via natural selection produces optimal solutions to these kinds of problems. I don't want to talk about that. Forget this slide. It's complicated. Phenotypic plasticity becomes an issue because individuals can behave differently depending upon what the environment is. So these individuals would all be individuals of a single genotype, but as spring temperature changes, the date at which they lay actually goes down. And so you can actually look at actual data for this. These are all, this is the mean laying date. And as you can see, the temperature that you're experiencing in the springtime affects the laying date. And so assuming that this population of birds is not particularly genetically diverse, these individuals are responding to seasonal cues that affect when they start laying eggs. I don't know if you guys know or not, but I raise chickens. I have backyard chickens in my backyard down here in Liberty. We're allowed to have 6 chickens. And one of the things that you'll notice when have any of you guys raised chickens like at home on a farm or anything? Yeah. What happens in the winter, Elizabeth, with egg production?
It decreases.
Yeah, it decreases. So I guess about mid-November, our chickens just stopped laying eggs. And I'm so disappointed because I keep feeding them, I keep watering them. They live in a heated chicken house. I mean, my chickens live better than I do, I think. And they haven't produced an egg all winter long. And just a couple of days ago, I went out to the chicken house and there was a single egg. And it's the first egg that I've seen since like last November. And I've been threatening them. Every day I go out and I say, if I don't see some eggs, I think we're looking at some chicken soup. It has no effect on them whatsoever. But the thing that is happening is they have sensed the change in day length. The day length is getting longer because December 22nd, 23rd, that's the winter solstice. After that point, we went from the days getting shorter and shorter to the days getting longer and longer. And they're sensing that. And as a result of that proximate cue, that seasonal cue, they're now starting, well, one of them is starting to produce eggs. I still need to have a talk with the rest of them. So environment affects what the phenotype is. In this case, the phenotype could be number of eggs produced. But that phenotype is definitely prone to environmental controls, this interaction of genes with the environment. Commercial egg production companies, you know what they do to keep egg production up all year, don't you? What do they do, Elizabeth? You shook your head, yes.
They keep them under lights.
Yeah, they keep them under lights. So they don't ever let the chickens sense that the day length is changing. Now that also wears those chickens out over time. They don't live as long. They're terrible. They're in terrible shape when they finally do die. But the fact of the matter is, if you want to get rid of genotype by environment interaction, what you do is you get rid of variation in the environment. And this is a commonplace thing. This happens a lot with lots of things, physiological traits, but this can also happen with behavioral traits as well. And that pretty much covers kind of this introduction to animal behavior as a field. in terms of the way we think about it. We always think about animal behavior in the context of evolution shaping behavior. But we have to think about evolution shaping behavior within the context of, what is it that is being selected for? How does selection act? How are traits determined by genes? How do those genes get expressed in ways that affect behavior? How do those genes interact with environment? These are all things that we have to kind of think about simultaneously while we're thinking about behavior. It's a lot to pack in your brain, but you'll be better at thinking about behavior if you can think about all those things all at once. Questions? All right. In lab tomorrow, we are discussing the Lima and Dill paper, and we're going to be discussing the reading exercise that I put on the Moodle site for you. So we're just kind of going to go through that list of steps that I gave you for how to look at that paper and kind of just spend lab. And we won't be in lab the whole time, I don't imagine, because we're going to get tired of this exercise after a while. I'm sure you probably experienced that as you were reading it. So I don't think we need to meet in lab tomorrow. A lab seems noisy and echoey to me. And so I'll send you out here in a little while a different meeting space. The other thing that I'm going to do is I'm trying to find another meeting room, not because Marston was cold, but because the mechanical equipment in that room is really loud and I can't really hear you guys very well when you're responding. So I think we're going to move to a smaller space, probably over in the PLC, and we'll just hold class over there for the rest of the semester. I just need to check room availability, but I'll send you out an email here in the next couple of hours about a place to meet tomorrow and a place where we'll meet for lecture for the rest of the semester that's a little more pleasant. Mason, you're still in quarantine, so I'll send out a Zoom meeting request for tomorrow, so you can join us for Zoom. Well, actually, would it be better to just have everybody meet in Zoom from wherever you're at tomorrow for lab?
Yeah.
You guys going to be talkative? Okay. Well, would you rather just do that?
That would be
Or would you
fine.
rather meet in person?
Zoom with me, either one.
Okay, then why don't we just do that? We'll just meet on Zoom, since Mason, it would get complicated with Mason being away and us in person, because in order for Avery and Elizabeth to hear Mason, everybody's computers have to be up in volume, and then we get feedback from the microphone, et cetera, et cetera. So why don't we just do that? So come prepared to chat. I want you guys to be doing most of the talking and me doing most of the listening as you go through this. One of the things that I think we do a pretty good job of with our graduates here at William Jewell College is we do a pretty good job of teaching you how to write. And I think part of the reason we do that is because we concentrate on it in a number of our classes. And this is certainly one of them. I got a Facebook post from a former student, and she is in graduate school, and she had just written her first lead author paper in graduate school. And she had handed it to her PI, her major professor in the lab that she was working in. And the major professor read it and had really relatively few edits, didn't really have to trim it down at all, thought it was in pretty good shape, et cetera, et cetera. And she wrote me because I think I was the one who kind of harped on her writing the most when she was an undergraduate, to basically thank me for being such a pain in the ass about writing. Tomorrow, be prepared to be talking, not so much about the content of the paper, the science of the paper, but about the writing of the paper and how it approaches the content in a sense. I think there's a lot we can learn about writing from reading these things, not from the science standpoint, but from the writing standpoint. All right. Any questions? All right, then I will see you guys tomorrow at 10:45 on Zoom, and I'll send you out a Zoom request. All right. Have a good day.